Well, while that's going on, just to give you a background, we have enjoyed working with the third cluster and COHA over the last few years. We've had to do some very dramatic adaptive management in our research program because of things that were out of our control. Certainly, one of the major things was having to close down a lot of our research because of restrictions associated with the pandemic. So we moved along quite a lot, and we're planning more work in the future as Charlotte completes her master's degree, which is now coming along nicely. So here we go. We're going to talk about the hollow structures of plants. And this is an area which is interesting because when we started working on this, and I started working on it, it was very difficult to find anybody who had paid attention to the pubescence and the flowers and the stems and the fruits, and even algal structural structures and fungal structures and lichens in terms of the endomicrometeorology within the hollow structures. And we've come to the conclusion that this is a lot more widespread. Next slide. Then, okay, here we go. That is an area which has been neglected. We know a lot about the micrometeorology around plants, so atmospheric movement, wind and convection, etc. We know light conditions are important to consider. Temperature and heat, of course, are very important aspects in agricultural climatology and crop production, as is humidity and precipitation. But nobody seems to really have paid much attention to what micrometeorological conditions exist within plant parts. So around them, yes, but within, no. And we have coined the word endomicrometeorology to discuss this, and it's not been studied very much except within flowers. So the next slide is one of our first attempts to model the process, looking at hollow stems, particularly with solar radiation coming in. Some is reflected, some is absorbed, some is transmitted because the hollow stems are translucent, and the absorbed radiation is then conducted, and some is lost out of the into or out of the cavity in the stem or the other hollow structure, and some is transmitted, and this adds heat to the lumen, as does the radiant energy can be also reflected and absorbed. So it's a complex biophysical thermodynamic process which is going on, and we've likened that to the greenhouse effect. And of course, the greenhouses also liberate heat, so it's emitted and it's also conducted away, and some by the phloem and the xylem, and some used in photosynthesis. So it's quite a complicated process. And we have modeled that, and we've been watching the literature for people who have shown an interest in this, and quite a number of people have questioned us. So we'll be bringing up where we are with this. So the next slide. So one of our objectives, and this is what we'll be focusing on today, was to review the biodiversity of hollow structures in herbaceous plants. This was one of the things that we put down as an objective with COHA, and it's much more widespread than is generally known, and it stems from plant pubescence, what's going on inside hollow flowers and buds, inside a few fruits of quite a number of different kinds, and bracts and flowers and reproductive structures, and within stems. So we have submitted a paper to the top journal in Canada, Facets, an NRC journal, and it's in review at the moment, where we haven't heard back anything. It was submitted back in late November. So the diversity we'll be going through, and we've referred to this as the structural and functional aspect of this as heliocaminiform. And the Roman Emperor Hadrian built a heliocaminus in which he would bask in the sun that came in through the windows. So in essence, the COHA project has opened new doors to botany, meteorology, and ecology. Next slide. And this gives us an idea of, sort of the pubescence. It's hardly been studied as a heat trapping, except in willows, particularly Arctic willows, in which female catkins are warmer than male catkins in the sunshine. In shade, there's no difference with the ambient air. And then some Arctic plants, such as the one in the middle, which is a lousewort, as Pedicularis hirsuta. You can see why it's called hirsuta, and heat accumulation on that. And there's a little bit of experimental work has been done on shaving those hairs off. And in the cactus, despite the fact there are lots of cacti with the pubescence, it seems it's been mostly associated with UV protection rather than with temperature and temperature control. Next. We started work with on our COHA work with looking at hollow flowers and particularly snapdragons. And this gives us an idea of various colors of snapdragons in a greenhouse at the University of Guelph with the instrumentation shown. And with the micro thermocouples that we applied, we have connected to the temperature recording device. We found that inside the antirrhinum, or the snapdragon flowers, we got temperature excesses up to 4.5 degrees centigrade. Next. And outdoors, this amount was even greater, up to 7 degrees centigrade. And we did some comparisons of floral colors, and the results were not as expected. And we've got more questions to raise. We've raised this before with this particular group as to whether pale colors should be warmer from greater micro greenhouse effects, or should darker flowers be warmer from more solar absorption. And we just don't know. We have not been able to follow up on this because of the constraints of the last few years. Yep. But to give you an idea, this is some work that I did way back in 1965 to about 1968 with the translucent syncales of Silene, then in the Caryophyllaceae on the right. You can see they look like Japanese lanterns, and they're up to 6.2 degrees centigrade above ambient under sunny conditions in the high Arctic. So this was done at Lake Hazen up on northern Ellesmere Island. And for Pedicularis capitata, another lousewort, up to about 4 degrees. And the orientation of the flowers does play an effect. Those that are parallel with the sun's rays don't heat up as much as those that are flat on to the to the sun's waves. So this is important in the high Arctic, particularly with 24 hours of sunshine. The sun just goes round and round in the sky during the time when these plants are blooming. Yes. One of our model plants that we have used under the with COHA is not an ornamental. That's the standard milkweed that we're all too familiar with. And we noticed that the fruits are hollow inside, and so if you pinch them between your fingers, you get some palpability there. And we found, much to our surprise, that on a nice summer day, we would get up to about 13 degrees centigrade warmer than the ambient air surrounding the follicles or the pods as they were blooming. So this was far more than we expected, and we're analyzing the data now to see how it might model out, and it will be a chapter in Charlotte's thesis. Next. We also looked at ground cherry fruits, and ground cherry fruits, as you know, have a sort of a hollow syncalyx that surrounds the fruit that one eats. And again, the daily temperature log of developing physalis fruits inside that envelope, then up to 4 and 5 degrees centigrade, warmer than the ambient air temperature. And the Chinese group has looked at that in different species of physalis and have recorded some of the same sorts of things. So we're looking at that, and again, a lot of analysis of the data to do. Our labs have been pretty much closed down because of the of the pandemic, and it's only recently we've opened them up again. So next one. And another one, in Love in the Mist, which was growing in my front gardens, beautiful blue flowers, and the fruits that are very hollow and have a mean temperature excess of about 5.5 degrees centigrade above ambient under full sunlight. And when it's cloudy or at night, that effect is abolished. Next. And the same goes with Echinocystis lobata. That's the prickly cucumber that we're familiar with. It blooms later in the summer, and on a sunny day, the incident light, a high amount of incident light, we got over 8 degrees centigrade temperature excess inside the fruit, again hollow, and on cloudy days, that's all but abolished. So we haven't been able to follow up on this as much as we have with others, but nonetheless, it's an interesting hollow fruit from a point of view of its heliocampiniform functionality. Next. Next. One of the things that struck us as we were doing our review, there are some things that haven't really been paid much attention to. And as we know, when ears of corn are starting to mature on the corn stalk, they feel as if you can sort of feel them with your fingers, and they feel hollow to touch. Temperatures within those ears of corn seem not to have been measured. And this is one of the things we hope to do this coming summer, is to use a cornfield, sweet cornfield at Strom's farm, just outside Guelph. Amy Strom and her family have been most welcoming to us. And similarly, the boot stage in the growth and flowering of grasses, cereals and sedges, these are presumed to function in protection, but they may also work very nicely in heliocampiniform heating. Next. We also looked at stems, and some of this data we have presented to you before. This is the paper white narcissus stems in a greenhouse at the University of Guelph. They are hollow inside, and when you look at them inside and cut them open, they look as if they're wet inside, but it's not. It's that they're shiny. And next slide shows amaryllis, which again is the same sort of thing. We don't, we had a lot of bulbs. We were given a lot of bulbs by a cooperator down in Niagara. Unfortunately, we weren't able to use more than a few of them in our initial experiments, and with the closure of the University of Guelph greenhouses from as a result of the pandemic. Next. Dandelion is similar. Inside the dandelion stems, which elongate extremely rapidly in the early part of spring and early summer, and have the same growth spurt associated with hefting the fruiting structures above, mean flower temperature with an infrared camera, and we'll talk a little bit about the infrared cameras, about 4.5 degrees more than ambient air, and the temperatures within the stems with a thermocouple then 2 to 5 degrees centigrade. So again, the same sort of thing, but the condition is abolished, as the graph shows, during cloudy periods or at night. So interesting thing from a point of view of weed scientists and the rapid growth and maturation of dandelions in the early summer. Next slide. Gerbera jamesii was our original major focus for our project, and we were made very welcome by Van Geest Brothers greenhouses in Grimsby, Ontario. And this shows the setup we had at Van Geest, and one can see there a box in the foreground which has instrumentation in it, and then from that instrumentation the thermocouple wires were extended into the stems and into the flowers of different Gerbera varieties. And really, we were very fortunate in that the personnel at Van Geest were able to look after the project as, and we were able to just run back and forth from Guelph to Grimsby 2 or 3 times a week to monitor everything. But that all had to cease during the pandemic. Next. So in looking at that, we thought, we should use a thermal camera, an infrared camera, and we got that from a company called FLIR, F-L-I-R, and we realized that before that we weren't getting similar results with the thermocouples and with the thermal camera. They're measuring 2 different things. The thermal camera is measuring the surface temperature, and the thermocouple measuring then the general temperature on the surface and around the flower. So we collected data over 4 days on 5 different varieties of Gerberas. Next slide. And we ended up with these sort of results. So the thermal image in a flower of Prestige cultivar, 5 degrees centigrade above the surrounding vegetation, and temperature measurement by thermocouples higher than that. So a little bit of difference, and the next slide shows what happens when we start to calibrate the thermal camera against the thermocouples. It's a linear relationship, which makes things fairly simple from a major point of view, from our worries about what are we actually measuring. And so we were able to use the calibrated thermal camera parameters to quantify and adjust our recorded temperatures by thermocouples. So that was that, I think, is a useful thing from a point of view of growers who might be interested in using thermal cameras to measure the environmental conditions in their greenhouses and outside, for that matter. Next. We also moved out into field crops, because that way we were able to get a team of people into the field, and they could stay 6 feet apart according to social distancing. And so we were looking at squash and looking at radiation, the amount radiation load on squash plants, relative humidity, and the temperature data loggers, such as we had already used in the greenhouse. So that gives us a field crop that was used at Strom's farm in nearby Guelph. Next. Again, we find big differences, particularly in sunshine within the petioles of the flowers, and that continues through into the peduncles of the fruits as the fruits are maturing, and seems to be an important component in the growth and maturation of the flowers and the fruit. And the differences between temperatures are different. This is 4 all done together, and some of them are more shaded than others by the leaves of the plants and so on. So the next slide, we looked at the nectaries, the ongoing studies in the flowers. And this is because of collaboration, which we started a couple of years ago with Dr. Robert Raguso at Cornell University, and looking particularly at the flowers in terms of their attractiveness to pollinators, because they have to be cross-pollinated, or they have to be pollinated because the flowers are of 2 different sexes. So the pollen has to move from the male flowers onto the female flowers in order for a pumpkin fruit or a squash fruit to develop. So we found the nectaries seem to generate their own heat. So it's probably a metabolic effect brought about by the nectar being converted from starch as the flower opens. And the flowers are only open for a day. So it's an important period of time from a point of view of pumpkin and squash production. Next. So in doing our review, one of the things I started looking at, and Charlotte started looking at, is what else was there out there? And we thought, okay, model plant parts. And I remember walking around in the intertidal zone in the Maritimes, and also on the West Coast, seeing the bull kelps. And they have then these gas-filled translucency, buoyancy compensator organs on them. And we wondered if those too might function in a heliocominiform way. But nobody, as far as I know, has looked at that. We do know a fair amount about the gases which are in there. They're just the common atmospheric gases, and they seem to be important then in buoyancy. But temperature has not been measured. We're hoping to get at that this summer, but we'll have to wait and see. Next. And then I was thinking about morels. Morels are an interesting fungus in as much as they don't grow in the shades of forests as so many of our fungi do. They're more they grow within open habitats and grow very rapidly once they start to come above the ground, and they're hollow inside. We don't know if anybody's looked at the temperatures in there. And also lichens. In the podetia of lichens, we have measured temperatures of a couple of degrees warmer than ambient air temperature. But again, it hasn't been looked at very much, and it's been a matter of, have we had the opportunity to get into the field to do those sorts of things? So the next, and is to just thank you very much for your attention. I hope you found it interesting. We've got a long way to go in terms of sorting things out, and we hope our review will be accepted fairly soon, but we're still waiting. And we're expecting to produce quite a number of shorter and longer papers on the different features of this micro greenhouse effect that we're seeing in hollow structures in herbaceous plants, and possibly other things that would have been years ago referred to as plants, such as fungi and lichens and brown algae. Thank you.
Great. Thank you, Peter. Excellent presentation. We do have one question. We are a bit behind, but it's all good. You, Ben, go ahead.
Peter, this is very interesting, very useful data. Just one question is that, within the flower, when there is sun, and within the flower, the temperature were higher, but in the night, it's actually lower than the ambient temperature. Do you have any explanation for that? It's just intriguing.
Yes, we would suspect at night that evapotranspiration is still going on, and therefore one gets a cooling effect from that. The other interesting thing is that some horticultural literature on chrysanthemums has indicated that the greater the amount of difference between nighttime and daytime temperatures, the faster the growth of the stem. I don't know that there's much published on that, but it would be of interest to be able to measure that sort of thing when we can get back in greenhouses and work with people. There's a question on chat from Jesse. The Arctic pollinators take advantage of the warmer flower temperatures, probably because the nectar which they're after, it has a lower viscosity at the higher temperature, so they can take it more readily. And also it's probably important in the maturation of the andrecium and of the gynoecium from a point of view of pollination in such a short growing season. In the Arctic work that I have done with the pedicularis and with the bladdercampions, as well as with some rosaceous and saxifragaceous flowers, we reckon there's a 25% gain in growing degree days as a result of the phenomenon. So yes, it's important for the pollinators as well as for pollination and growth and maturation in such a short period of time under relatively cool conditions.
That's wonderful. Great. Thanks, Peter, and thanks for the questions. It makes me think about it just makes me think about the pests in greenhouses and whether they're more attracted to the warmer temperatures in hollow stems and flowers as well.
That we don't know. It's a very interesting point. It certainly crossed our minds. But working with Van Geest, of course, they had their pest management scheme running the whole time that we were there. There was nothing. They gave us special treatment in giving us a good length of bench space with a whole variety of Gerbera flowers. On it, the Gerbera flowers, different Gerberas have different hollownesses of stems. Some are almost solid with rather pithy, others are hollow, others are a mixture of both. We were going to get at that in a more detailed fashion with Charlotte's work until we were cut off from access to really anything but field work because of social distancing. Charlotte, do you have anything to say?
No, I don't have anything else to add. I think Peter answered the questions already. So yeah, it's just interesting as well to hear some of the other researchers looking at temperature this morning. So just sort of like taking that in, as well as thinking how these projects could actually overlap in some of their importances. So that's one of the things that I would highlight as well. So thanks so much, Peter, and for everyone else as well.
Well, thank you, Charlotte. It's been great working with you, and we're still working together. And we should have a flurry of papers coming out. A number of the things that we've already published are already up on the COHA website. There's a video up on there from a year or so ago, and our other presentations to COHA and some webinars, etc. So please feel free to have a look at those and send us questions.